Secondly, I just want to mention that although we did announce that Dr. Triplett would be giving his talk live at the Oak Room, we had to make a last-minute change because he was exposed to someone with COVID yesterday, and we felt it was safer and cautious enough to have him present on Zoom, to which he has graciously agreed. Dr. Triplett is the chair of microbiology and immunology at UF. He has been here since 2003. His research activity has been on the infant microbiome and how it is affected by the mother's microbiome and affects the individual's neurocognitive and autoimmune functions as late as 20 years later. His topic today is merging research, teaching, outreach, and administrative goals in a single microbiome research project. And this seems to me like he will give us an overview of the subject and also talk about the gut microbiome in infants, right? So thank you, Dr. Triplett, and it's all yours.
Yeah, thanks for having me. I did a switch of topics a bit. I'm going to talk about actually the saliva microbiome in university students, but I hope you'll find it entertaining, and I'll see you like that. So it's the first time I've given a talk on this subject. We just started this a few years ago. But what I'm talking about is a project that we've used to have multiple goals, both goals for the department, goals for students, and goals for science. So what I'm going to be talking about is what we call the Great Florida Spitting Contest. And I'll tell you why it's called that in a little bit. And I'll describe how this fits the mission of the department. And what we're trying to do is provide a microbiome-based platform, a multi-omics platform, if you will, to provide research experiences to as many of our students as possible. And we have a lot of them. We have about 1,000 majors at the undergraduate level. We have about 750 master's students and about 60 PhD students. So our missions are, as a department, are to grow research efforts at all levels, grow the graduate program, increase enrollment and retention, decrease time to degree, increase diversity at all levels through online education and growth, integrate these endeavors whenever possible. And these things are not mutually exclusive. They can all be done at the same time. So one of our issues is with all the students we have, it's difficult for us in our laboratories to provide research experiences. We just don't have enough space or enough mentoring power to accommodate that many. However, they need it because the research shows that research experiences are the best way to increase retention at the undergraduate level in STEM. Our Great Florida Spitting Contest allows us to provide such experiences easily. Our online programs increase our diversity. So we're perhaps one of the few STEM departments in the nation that has offered all of its lectures online, and it has done so for years prior to the pandemic, starting in 2011. And our purpose in doing this was manifold, but one of them was to increase diversity, and that's worked. The diversity of our student population now exceeds the diversity of the state, which we're quite proud of. Anyway, the project I'm describing today has assisted in that. So our goals were to develop a microbiome project that could provide students with research experiences through courses delivered online and to generate publishable data on a novel microbiome project. So we thought long and hard about how to design such a thing, and we wanted students to use their own samples. We wanted to be IRB approved so we could publish it. Students would be volunteers and provide their own samples, and they would analyze the data and actually write many papers at the end of courses. And we decided to study saliva rather than stool for this because we thought it would be far easier to get saliva from college students than would be stool. And that's turned out to be the case. But like any research project, it has to have an overarching question. And so, pardon me, the overarching question for this work was, to what extent does human genetics drive bacterial composition in the mouth? And so we use saliva as a source for both human DNA and bacterial DNA, from which we would get an assessment of human genetics by NIP analysis, similar to what's done by Ancestry.com or 23andMe, a very similar procedure. And we'd analyze bacterial DNA and bacterial composition by amplifying and sequencing the common taxonomic character for the bacteria, the 16S RNA gene, which you must have heard about already this semester quite a bit. So to answer this question, we have to consider any confounders. So what might get in the way of us understanding the extent to which genetics drives bacterial composition of mouth? Well, it's fairly obvious, I think, that diet must affect composition of mouth. Diet serves as the food and energy source for the microorganisms in your mouth. And another confounder that surprised me when we started this is actually depression, because depression is associated with periodontal disease. As you know, periodontal disease is caused by bacteria. So we had to keep participation by students simple. To get a good enrollment in any human cohort, you have to keep the thing simple. So all the student participants were volunteers, none are paid, and to get high participation, we decided on these principles. First, we use saliva rather than stool. Second, to estimate diet, we use a survey, not a lengthy multi-day diet diary, which would be too complex for them to do, and too distracting from their studies, frankly. And we also need a simple assessment of depression, not a standard medical mental exam. And other surveys were added later for this, but all the surveys we use in this work that students complete are simple. I'll show you a couple examples. They're published, they're validated, and they're widely used. And that, of course, is done to reduce reviewer criticism. So how do you measure diet? What we chose was a diet questionnaire called the KidMed survey. It's a 21-question survey from Spain, and it's been validated by nutritional analysis. And what it essentially does is assess the level of compliance a subject has with the Mediterranean diet. And it's designed for young adults in Spain, and we thought it should be translatable to students here. And here are the questions asked. So each one of these questions gets at the extent to which a person is complying with the Mediterranean diet. Do they eat fruit often? Do they eat cooked vegetables? Do they consume fish? Do they eat a lot of fast food, which is negative in the survey? And you'll see the other things there. And you add up these points for each of these questions, and you get a KidMed score. And it's considered, if your score is above 8, you have a high compliance with the Mediterranean diet. If it's poor, you get below 3. So we used the KidMed. And as I said, depression is associated with periodontal disease, which has been known now for 70 years. So it's well documented. And we do see occasional bacterial pathogens in the saliva. So how do we assess depression easily? And so I went to the Wellness Center, and it was suggested to me that we should use the PHQ-9 survey, the Public Health Questionnaire number 9. This is validated in 2001, published, and it's a simple 9-question survey, and here are the questions. And it's a matter of whether or not you've experienced these things at some point during the last 2 weeks, whether it be little interest in doing things, feeling down, depressed, or hopeless, etc. So the total score from this survey, but the points given for each factor here, give you a total depression score. And number 9, the 9th question there, is the one that deals with suicidal ideation. And I'll get to that in a minute. That is, is a student having are they thinking about self-harm, basically, over the last 2 weeks? So it's been shown in the literature that this survey correlates well with mental exams. And this is the data showing that. I don't think I need to go into that, except to say that for a minimal score on the PHQ-9 score seen here on the left, you're far less likely to have a depressive disorder. Whereas if your score is very high, you're much more likely to have a depressive disorder. So we used that. And question 9 became important to us. I'll explain why in a minute. But when you ask the question, have you often had thoughts that you would be better off dead or hurting yourself or in some way or hurting someone else? And how frequently do you have those thoughts? And when someone answers this positively, either 1 through 3, in those responses, we are required to refer them to Matter We Care, the Dean of Students Office. So hence, the GFSC has 4 measures. We measure the saliva bacterial diversity. We measure their genetics through a SNP array. We determine their diet as it relates to the Mediterranean diet, and we do a depression survey. And prior to the pandemic, that is prior to February 28th, 2020, we had signed up 489 students. On that day, saliva collection was suspended and brought back about 18 months later. But the samples I'm talking about today were all collected prior to the pandemic. We now have a large collection of samples since the fall semester of 2021, but over 800 student volunteers to date. Now this thing is IRB approved, and we restrict it to students who are taking one of our courses, or if they're one of our majors, they have to be 18 years old or older. And the identity of these participants are not known to me unless they voluntarily tell me, but I never ask, and I don't need to know. I just need to look at the data. The only folks who need to know the identity of these people are when they score high on the depression score, then we have to refer them to the wellness center, and that's done by our study coordinator, not me. So why in the world is this called the Great Florida Spitting Contest? Our vision early on was to collect samples from several Florida universities and colleges to make this a statewide effort. And that's still our vision. We still have to do that. We haven't expanded that way yet, but our hope is to eventually make this a contest indeed. So we just recently, last August, published our first paper on this large data set. And this was all done by a PhD student. Now she's graduated, Angelica Ahrens, and she had a lot of assistance from undergrads, particularly Diego Sanchez Padilla, and others in our department. Other faculty helped as well. And we decided to have a focus on suicidal ideation. And why did we focus on that? Well, suicide is the second leading cause of death between people of the ages 10 and 34 in the United States. And it's a worldwide problem. Across 108 countries, 24% of college students have harbored suicidal ideation at some point or another. So it's a big problem. And the causes and solutions for SI, as we call it, are understudied given the magnitude of the problem, particularly on college campuses. When we survey students on this campus, generally about 12% report suicidal ideation. So it's something, on my view, we can't hide from. We should study the problem and try to solve it like we would any other problem. And we do consider this to be the most novel aspect of the GFSC. And I'm interested in this myself because in 2015, our department had a graduate student commit suicide, and it was just devastating to us. And as is often said, none of us saw it coming. And I thought, well, might there be a way to study this? So we're doing our own part to look at this problem. So What I can tell you is that the Mediterranean diet really plays no role in whether or not a student has SI. It plays a role in depression, which I'll get to in a minute, but not SI. But that may be because their numbers are too small. You do see the trends there, that greater mental health with a higher compliance, but with our current N, it's not significantly different. However, consumption of nuts and sleep was associated with SI in women, but overall in depression was also associated. So we can now urge our students to get sleep. I think that's a huge problem, and improve your diet if you can. What we're doing in this department is having a serious discussion about how we can reduce the number of high stakes assessments we give our students. We think that causes a lot of unnecessary stress. We want to move to lots of low stakes assessments so they're less nervous. And this came to us when we noticed that there was a huge spike in depression in the month of October. And we think that's because that's when the first set of exams set in and students become more and more nervous. Anyway, I digress. So then we looked at bacteria that are associated with SI, and we found some that are higher in SI, such as Bacteroides dorei, which, by the way, is a bacterium associated with type 1 diabetes in children, Veillonella atypica, Pseudomonas tisacea, Klebsiella oxytoca, and Streptococcus. So these are higher with SI. However, we saw more bacteria, and the effect was more pronounced amongst those bacteria associated with no SI. And what is commonly the case in the microbiome, and you probably heard this earlier this semester, it's hard to find bacteria associated with the condition of interest. What you do find is bacteria associated with health that are lower numbers than those with that condition. So it's typically the microbiome moving away from a healthy state to an unhealthy state, and that unhealthy state can take many forms. So that's true in this case too. And we also looked at bacteria when we controlled for sex or gender, diet, and sleep. And even then, we still see bacteria associated with SI either positively or negatively. So in genetics and SI, we saw a surprising result. Up until our paper was published, there were no reports of HLA genes in the human being associated with depression. Now, the HLA genes are the human leukocyte antigen region. It's on chromosome 6. They're quite variable, and they play a major role in human interactions with bacteria and viruses. So I became interested in this with type 1 diabetes and celiac disease. We published on this. It's well known that the MHC class 2 alleles, DR4, DR3, DQ8, et cetera, that these are commonly associated with autoimmune disease. And we found here that with respect to suicidal ideation, we see that some of the same alleles that are associated with autoimmune disease are protective from suicidal ideation. That was a big surprise to us. And what was quite striking to us was DR4. DR4 is a gene associated with both type 1 diabetes and celiac disease. And we found in students that had that allele, there's some 63 or so, none of them had SI. And typically that number is 12%. So that was interesting to us. I think a very important result for this paper. And there's more on genetics too. We haven't looked at this exhaustively yet for statistical reasons. That's another 15-minute story. But my student did look for genes that were associated with SI in the literature or with attempted suicide. And she found this one SNP that's within an intron, within the KIAA 1549L gene on chromosome 11, that is codes for a protein enriched in the brain. And this particular one base region of this gene, when that allele is a G, ATCG, the 4 letters of the alphabet of DNA. When that's a G, this gene is associated with attempted suicide. So that was published. So we asked the question, is this SNP, this one base region, is it associated with SI in our cohort? And the answer is yes. That if a student has the G allele, so you have 2 alleles for any base in the DNA, and if either one of those is a G, your chances of SI go up 2.6-fold. So what we're seeing is there's a genetic connection to suicidal ideation, which has not been studied much at all. And it's what our work is getting toward is perhaps a way of screening students who are at risk, whether it be through their sleep habits, their diet, through genetics, through surveys, whatever. Can we identify students at risk early and intervene early before they have problems? Then the question is, are there any bacteria associated with this SNP? Could it be that this SNP somehow controls the microbiome, controls its composition, such that it has an adverse effect on mood? And what we found was that in those students who did not have that harmful allele, G allele, they had a much higher abundance of Alloprevotella rava. That is, healthy students without SI had this bacterium if they had double A, the 2 alleles of A in their genome for this particular SNP. If either allele was G or both were G, it had much lower Alloprevotella rava. So we thought to ourselves, what's this mean? Well, it does appear that if you have AA at this particular SNP, you're likely to have more Alloprevotella rava, and it appears to protect against SI. Now, why might that be? Well, it turns out that Alloprevotella rava is fascinating in that it ferments glucose to succinate. So whenever we see a bacterial association with some phenotype of interest, we try to dig in and understand its physiology. What do we know about how this bacterium works, and how might that relate to any human condition? And in this case, Alloprevotella rava is one of the rare bacteria that ferments glucose to succinate. So I thought, well, what's that got to do with anything? So I looked in the literature, and it turns out that succinate has been known since about 1947 to improve oxidation in the brain. And succinate has been used for decades as to perfuse people with succinate to reduce brain trauma, and it works. So here we have a bacterium in healthy people in the mouth that probably makes succinate. And you can imagine it could make its way through the brain. And that may be improving the mood of students. So could it be, and this is one of our long-term goals, could it be that a bacterium like Alloprevotella rava could serve as a probiotic for mental health that you could deliver in chewing gum or mouthwash or whatever, and hope it colonizes? The problem, however, may be that if you have the wrong genotype, for whatever reason, this bacterium won't colonize. But perhaps with persistent application of the bacterium like a probiotic, it might work. So, as any good University of Florida faculty member would do, I immediately went to UF Innovate and said, maybe we should patent this. And so they filed a provisional patent. So the summary, this is the summary of the first paper, but there is so much to do. We now have double the size of the cohort that we had here. And let me just give you just a thumbnail sketch of what more we could do. We haven't looked at the overall connections between bacteria and depression. We need to look more at genetics and diet. We do, by the way, we do see connections between bacteria and depression. We just haven't published that yet. And on genetics, what we're learning is that you are what you eat what you are. It isn't that you are what you eat what you are. There's a lot of genetic predisposition to your palate. And I think that's going to be a great story in of itself, just publish that. And we need to look at genetics and depression from a different insight, perhaps, that has been studied before. But in past studies, no one's found this HLA connection. So we thought it's worthwhile for us to take a look as well. And as our cohort grows, we'll be able to say more definitive things about that. Now, we want to get back to the original question. How does genetics affect bacterial composition in your mouth? We know that about a few bacteria already. Alloprevotellus rava is one of them, but we have 940,000 SNPs to look at, and I only presented one today. So there are many more we need to investigate and learn just how the genetics controls what's in your mouth. And you can imagine if genetics is also controlling diet, that's having a big impact on what's in your mouth as well. We're looking at this data set to see if it can be used to predict celiac disease. Celiac disease is a condition where people have an allergy to gluten, a common protein found in wheat. And there are lots of complicating factors if you are sensitive to gluten and it's not diagnosed. And this is a disease that often goes underdiagnosed and causes lots of problems in folks. So we want to see, given what we are looking at, and by the way, celiac disease can be associated with headaches. Can we see anything in this data set that might allow us to predict undiagnosed celiac disease in subjects, which could lead to mental health problems such as headaches? It turns out that we have 14 people in our cohort who are all gluten-free, and none of them have headaches. So the cohort needs to be bigger. We're getting there, but I can keep you posted over time. Another thing we want to do, at least with respect to diet and depression, is kind of understand cause and effect. And the only way to look at cause and effect in those circumstances with the microbiome is to follow these associations longitudinally over time. And fortunately, we had 2 or more samples from at least 50 students now. Maybe it's closer to 60 now. And as that cohort grows, we can look more and more at cause and effect. So with that, I think what I want to say to you is it is possible to design a research project that meets administrative goals, teaching goals, research goals, and all sorts of things, and still be interesting. And the NSF has funded a bunch of the supplies and things used in this work. So we now have a large multi-omics data set that is being used in 5 courses to provide all the students in those courses, that might number about 1,000 a year now, with an undergraduate research experience. That's a lot more than we can provide in your laboratories. This is developed into an important research project that has already generated 1 PhD degree, and I think will generate more soon. This project serves as a means of outreach to other institutions. We've got 5 other institutions in Florida interested in working with us on this, including University of South Florida, University of Central Florida, Hillsborough Community College, Valencia College, Miami-Dade College, et cetera. And we hope to expand this further. And one of the reasons to expand this and get participants from more universities is a much higher number of students participating. We can get our results to be far easier to validate statistically than what we can do today. And we're about to look into how this project is helping our enrollment, our retention, our undergraduate and graduate programs. And of course, we want this to lead to more significant grant support. So it is possible to design something that can have a lot of objectives. And I'm sorry it's kind of a brief talk, but I'm happy to have lots of discussion. Stay here as long as you want, and let's brainstorm. So thanks for having me, and I'll close there.
Thank you, Eric. I can't wrap my brain around this tying up the mouth bacteria with mental disorders. What did you say? Ken is here.
Hello, Eric. Good to see
Is
you
that
again.
Ken?
Yeah.
Good to see you, Ken. Thanks for coming.
So the question I have, and is to get my head around this succinate issue. Does that imply that the succinate concentration, let's say in the blood, is higher in these people?
That's what we'd expect. That'd be worth measuring. We don't collect blood, but maybe we should. That's a good idea.
Because presumably, if it's going to be higher, it's got to be higher in
Yes.
the brain in
Yes.
order to accomplish
Yes.
what you're talking about.
I don't think these bacteria pass the brain. We did work with Charlie Wood showing that it can happen.
No, that's why I'm suggesting that.
Yeah, right. That's a good idea. We should look at that. We'd have to hire a phlebotomist, but it probably might be worth doing.
Yeah, anybody,
Good
it's
idea.
easy to get a good phlebotomist.
Yeah, right.
I guess
Holly's got-
the other question I have, Eric, is how challenging for you in this study was the IRB? Did they give you a lot of aggravation?
We don't have that kind of time, Ken. But yeah, it was difficult. But I think we've reached an agreement now on what to do. But it's hard to. They're very concerned about us looking at depression issues. So the concern is this: that, as you know, the rules at UF are, if you know something about a student being in some kind of difficulty, you have to report it. So they say, if you know something, you say something. And you have to say something to the Dean of Students office. They already know the depression rate on campus is very high, about 28%. The SI rate we find is about 12%. So if we surveyed every student on campus, which I think should be done, that's another story, they would be inundated with people they would have to serve. They just don't have the staff for it, which is kind of sad. So I can't blame them for wanting them to limit or study and perhaps not even do it at all. However, at the end of the day, I think they agree, it behooves us to study this problem. And we encourage people to work with us. Anybody who wants to join us is welcome to make more progress. But yeah, we had some criticism at first, and we got through it.
Thanks, Eric. I really enjoyed your talk.
Thanks for coming, Ken. Good to see you. Wish we could be there. Wish you could be there in person.
All right, we have a question on Zoom. Holly, go ahead.
Yes, very, very interesting. I was wondering if you're doing any collaboration with the Department of Psychiatry. This is a two-part question. And the other is, as a veteran myself, the fascination with what you're finding about depression, wondering if, and diet, when you're checking diet, in terms of like veterans or actually active duty, utilization of MREs, which is all they get in the field, and what a simple test in a way to be able to maybe determine some differences in what we see in varieties of depression and PTSD in veterans.
Oh, we're very keen on that. So we submitted a grant to the Army, to DARPA, about 2018. We called it, I forget what our title was, but it was to look at these very things in soldiers at Fort Bragg, particularly the airborne. And my grad student at the time, Angelica Ahrens, had contacts at Fort Bragg because she lives in Fayetteville, North Carolina. And we had a team put together, but they didn't fund it. It was pretty complicated looking back on it. I would like to revisit that. But what I am trying to do, however, it gets more complicated, I think, when the depression comes from PTSD rather than other sources. So I'm more interested, just because I think it's easier to look at depression in people who haven't suffered physical brain injury. But I do think the military needs to be looked at. I think it's a great point. And what we're going to try to we're trying to figure out is, you know, how big a problem is this in the airborne, and how is that related with the number of jumps and all that. So,
Well, I think across the services, I used to
yes,
practice in psychiatry, but across the services, there's a good deal, and I've been retired for a while, but of depression, and not diagnosed as PTSD, just depression for a variety of
yes, that's
reasons.
right.
So there would
That's
be,
right.
if you can get them to fund it.
Yeah, it's like anything else, right? Can we get the support? And early on in this project, we talked to psychiatry, and we got from them that they had the answers already, and they were too busy to work with us. So, and then we published our paper, and they saw it, and they wrote to me, and they said, you know, we could really improve your work if you just worked with us. And that wasn't a very friendly way to approach me, I thought. So anyway, we had already by that time established an association with the psychiatrist at Duke, who's NIH funded to look at depression in college-age students. And he's looking for interventions. So we think that's the best possible psychiatry collaborator we could have.
Great, thank you.
Okay, we have
Sure,
another
thanks for coming.
question. We have another question here at Okamic.
Sure.
Very fascinating presentation. And I can understand the challenge that you're faced with so much data to come to your conclusions in a very timely fashion. So my question has to do with, what are your strategies to eventually extend this into the general population, particularly those of us who are not students, but have some of the same issues with the biome, et cetera, et cetera. Obviously, that would be a huge undertaking when you're talking about from 800 to 1,000 to millions of potential candidates and subjects.
Well, I was trained as a plant physiologist, so I'm kind of out of my my expertise. However, getting more and more keen in the human problems. I'd like to get bigger, but I want to focus on one thing. I want to focus on the depression issue in the student body, and I want to nail that one. And then I hope that whatever we discover inspires others to look at a project of interest to them. I would like to do something in the military that'd be fun. The faculty here have asked me to study the faculty. I have no idea what depression rates are amongst the faculty. I don't think it's as high as students because they're not food insecure and their careers are kind of set. I think students have a lot more issues as early in adulthood. So my passion's on that problem. And I just don't think I have the time or the bandwidth to expand it too much more. But you're right, it should be done. I agree with you entirely.
Thank you, Dr. Triplett. I'm still kind of curious about the infant gut microbiome and if that's set up as the child is born and how that might relate to your study.
Yeah, so that's a different study we're doing, but I'm happy to say a few words about that. So we're working with Johnny Ludvigsson, who's a pediatrician at Linköping University in Sweden. He established a cohort of children in the late 1990s, and he enrolled 85% of the children born in a 6-county region of southeastern Sweden from 1997 to 1999. And that was about 17,000 kids. And he's continued to monitor them and to this day. And at 1 year of age, he collected stool from 1,700 of those samples. And at birth, from the pregnancy, he collected cord blood from every subject. A mammoth task. I don't know how he did it, but it's incredible. Anyway, what we're finding, so what we have are these samples at cord blood at birth, and we have samples of stool from 1-year-olds, and then we have medical outcomes over the next 20 years in those people. And we ask the question, are there any signs of future disease in cord blood at 1 year of age? And the answer is yes. That it looks like we see markers of inflammation already in the cord blood through metabolomics collaborators from Finland and Sweden that are predicted, are associated with future disease. And by disease, I mean autoimmune diseases such as celiac disease, type 1 diabetes, Crohn's, hypothyroidism, and there's another one I can't remember, JIA, juvenile idiopathic arthritis. And also neurocognitive disorders, and that includes ADHD and autism and sleep disorders. So we have all those outcomes. And we published a paper on those outcomes as on those factors that relate to future celiac disease. We've got a paper in revision now on type 1 diabetes. We've got another one about to be submitted on looking at all autoimmune diseases together. And do we see signatures of any autoimmune disease? And Angelica Ahrens, the author of the work I presented today, is writing a paper on neurocognitive disorders with respect to the 1-year-old microbiome and cord blood. And in cord blood, we look at metabolome. So that's all coming. And what's fast, so today, if you're trying to predict autoimmune disease, you look for autoantibodies. And they appear 6 months to several years before disease onset. But we're seeing microbiome changes at 1 year of age. We're seeing metabolomic changes at birth. And so it appears that these triggers occur very, very early in life, that the stage is being set early, far before we could likely see autoantibodies. The same thing is true with neurocognitive disorders. However, the changes in the microbiome that occur with future neurocognitive disorders is very different from what occurs with autoimmune diseases. They're both dysbiotic, as you've probably heard the word, but they're differently dysbiotic. So there's a world of work to do here. And now we want to look at stool samples at 2.5 years old, 5 years old, 8 years old. But as you can appreciate, it all depends on getting the money. Fortunately, I'm a small part of an EU grant that was just funded that will fund our work with this cohort for the next 6 years, at least some of it, but we have more to do. So I think the early infant environment is crucial. I think much of it is driven by genetics.
I think we'll have to get you back to talk more about that early infant study that you mentioned.
Yeah, I'll come back sometime.
Okay, so if everything is set at the time of birth, how could we change anything? Just by changing our microbiome? Would that help?
It might be too late by then because we're seeing these cord blood differences, right?
Yeah.
And some of it are environmental pollutants. You've probably heard of the forever chemicals. Those polyfluorinated hydrocarbons, those things are nasty. And I think they could be an important cause of future autoimmune disease in the environment. And genetics, of course, we know plays a key role. But what are the triggers? It can't be just genetics because not everybody's genetic risk has these problems. All right. George got a question.
Is there any link that has been shown, say, between microplastics and microbiome? Microplastics are such a big thing, and do they, you know, the nasty?
I haven't seen any, but I haven't looked hard.
Yeah.
I look harder at this, but there's so much to do. It's mind blowing. But I do hope that we can develop probiotics that can reduce autoimmune disease or reduce depression symptoms, that sort of thing.
So what are the probiotics that would alter the oral microbiome, the one that we want to have? What kind of? Well,
we think two big players are Lactobacillus and Bifidobacterium, particularly Bifidobacterium. That's a major player in the gut, and it's associated with health. And we just published a paper where we find that those at genetic risk for autoimmune disease are less likely to have Bifidobacterium in the gut in high numbers. And it varies with strain. One of the problems with often folks in medicine is that they think one strain of bacterium, Bifidobacterium brevi, is equivalent to any other strain, but strains differ, so it matters. And we're seeing great strain species differences between colonization of the gut, depending on your genetics. So I think at some point, we're going to have to personalize probiotics for a person's genetics. Otherwise, they won't work. And one of my other grad students, Megan Berryman, did a meta-analysis of 19 papers on clinical trials of probiotics. And in all of those cases, they didn't work. They didn't have an effect. And I think that's because they're not personalized. And that's where we need to go. Sorry, we had somebody waiting here for a question for a long time.
Wow, that's really getting complicated. Jordan.
Yes, thank you. Very interesting talk, but I did have a question on infant microbiome again. Something that I have felt is that the antibiotics that are given to newborn infants when they're put in the neonatal intensive unit, and this is done often, sometimes it appears not really necessary. And I know that that was the case with my second child. And she had horrible behavioral issues as an infant and up until she was about 4 or 5 years old. And the same thing when my grandson was born, his breathing was rapid. And that was the same thing with my daughter. They snatched him away and put him on IV antibiotics, and they're in the NICU rather than being held. But I really believe that that has very untoward effects on the infant's development. And I wonder if anybody's really looking into that, the long-term sequelae of this.
Yeah, we had a paper with, well, first, we are looking this at the Avis cohort in Sweden, and we don't see connections between antibiotic use during pregnancy or early in life with future autoimmune disease. I don't know that we've looked at neurocognitive disorders yet.
Mhm.
But we had a grant some years ago with Joe New in pediatrics, where he was keenly interested in that question because he's a NICU pediatrician.
Mhm.
And he says, we're given these antibiotics like they're, the just routine
Right.
care. And pediatricians are probably quite rightly scared not to give them because they want to prevent infection, right? So
Yeah.
it was difficult to get through IRB to test this, and probably rightly so, but he did. We had 92 infants involved, and I don't think we saw a connection between autoimmune disease and outcome, but they weren't followed that long. The N wasn't that high. So Joe was keen on expanding this to several hospitals, going up to a number of 300 or 400 kids, which was a great thing. He submitted that thing 3 or 4 times. He would revise it according to reviewer comments. Reviewers did them on different things, and he never got it funded. And it's really a shame. We did publish one paper, but we couldn't see any clear effects there. But I think it's because the N wasn't big enough.
Yeah, I was wondering more about neuropsych problems with that. I mean, my daughter was even given a lumbar puncture that I wasn't even told about. And when I wanted to just take her home, they threatened to call HRS on me. They were going to report
Gee
me to
whiz.
HRS. Yeah.
I'll ask Angelica, who's working on this with the Avis court, to see if she sees anything.
Okay.
But I don't think she has yet, but I'm not sure. I'll ask her.
Okay. Thank you.
But we're keen on the problem. We're keen on it. We just don't know. But it requires a lot more people working on it than just us. That's for sure.
Well, thank you. This has opened up a whole new field. I'd never even thought about it before. So good luck with your funding. I do know I served for several years on NIH study sections, and if I had gotten something of yours, I would have said, this is a fishing expedition. There is no.
That's right. That's right.
Yeah, really.
That's right.
But you have to start somewhere. Now that you have the baseline data, it should be easier.
I'm thankful, though, that both NSF and NIH, they're finally understanding that
Yes.
exploration is useful.
Yeah, you have to have the basic data to show, then it
Right.
takes off, and you have that now.
Right.
Good luck with that. Are there any more
Okay.
questions or discussion here? Anything else? If not, thank you so much. We really appreciate it.
Thanks for having me. Yep. Take care. I'll come back sometime. Take care.
Yeah, thank you so much. Before we end this session, I just have an announcement to make that Ophelia has asked me to make. Next Thursday at 3:30, there is a talk here on, it's called Learn More About the Plant-Based Diet, which is somewhat related to what we've done. And yeah, okay. And Linda D'Amico is the speaker? No. Oh, okay, okay. Oh, okay. So it's the practical ideas on how to feed your microbiome, which should be of interest to a lot of our audience. So it's next Thursday at 3:30 here in the Oak Room. Will it be on Zoom? No Zoom, so you have to come here. Thank you all so much for participating in this series. Thanks. Goodbye.